Welcome to this webinar that is part of a series about gene therapy and is a collaboration between EURO-NMD for rare neuromuscular diseases with the ERN for rare neurological disorders and the ERN for rare epilepsies, EpiCare, and also in collaboration with the European Academy of Neurology. We have the privilege of having with us someone who is an expert on certain complex and very minute details of the delivery of this kind of treatments. Professor Hildegard Büning, that she's a professor of infection biology, of the gene transfer, and a deputy director of the Institute of Experimental Hematology at the Hannover Medical School in Germany. She has served as president of the European Society of Gene and Cell Therapy and is the scientific secretary of the German Society for Gene Therapy. She's also a member of the board of the American Society of Gene and Cell Therapy. She studied biology and obtained a PhD at the Munich University, LMU Munich, and since then she is active in the field of adeno-associated virus vectors, with a particular focus on improving the efficacy and safety of these biological nanoparticles. So it's my great privilege to give the floor to Hildegard and to be very thankful for her willingness to share her knowledge with us. Thank you.
Thank you very much, Antonio. Thanks all of you. And it's really nice and a pleasure to discuss with you on gene therapy. And while you are listening to this talk, if we are just looking at us, so we can say that we are consisting of 3.7 times 10 to the 13 cells. And you might, depending on how you really look into this, we have 100 to 1,000 different cell types and approximately 25,000 genes. So that is we while we are listening here. However, what are we doing when there is a mutation, a change in our gene, which results in a defect in our organ function, or there's an accident and an organ becomes damaged, like a huge burn of our skin, or there's a malignant cell turning up and it's not recognized by our immune system, or even if we look at chronic infection. And for all these things, one kind of therapy is developed with the option of a curative therapy, and this is called gene therapy. And basically, if we just have a look at the simple monogenic diseases and have gene therapy here depicted in a nutshell, then we do have here the nucleus. I hope you see my cursor, with the mutations causing the malfunction. So the idea which we pursued long as an only option was that we add a genetic information. So for example, putting in a functional copy into the cell, hoping for a functional cure. However, now with the advent of gene editing, we are still left with the same condition. So we have a gene which has a mutation. However, now, and I hope my slide will turn now, because I know that I have a difficulty with these 3 slides. We have with gene editing really the option of doing a repair, and this is done as follows. So we first introduce near the gene mutation a double-strand break with the help of so-called designer nucleases. And when my slide is turning, then you see here one example of one of the first designer nucleases, the zinc finger nucleases, where you have a part of these nucleases being a protein which recognizes a specific sequence in the DNA, which you predefine, and then a nuclease domain which is doing the cut. There are other designer nucleases around in the meantime, and the most known at the moment, I think, is a CRISPR-Cas system, where we are much faster in changing the target sequence, because we are now relying on an RNA to find our target sequence in the genome for precisely put the nuclease function, instead of that we have to mutate the protein part of a designer nuclease. All in all, independent of how we generate the break, we have a DNA break at exactly the site, hopefully, that we would like. And then we provide a template having the correct gene sequence. And due to the cellular repair machinery, we now can really repair the defect. So if we look into gene editing, we can quote this as a more precise form of gene therapy. And this one, as you know, is there are a lot of clinical trials ongoing here, and it's more than that even. However, so independent, if we do a gene addition or gene repair, in principle, we do have two different routes of administration. So either we go for the ex vivo approaches, where we isolate from the cells, from the patient, our target cell, keep them in culture, modify them and give them back. One example are hematopoietic stem and progenitor cells, or for example, for CAR T cell technology, which we later speak on, the T lymphocytes. Or the other approach, which we call in vivo gene therapy, would be a delivery of the gene vehicle directly into the patient, either locally or intravenously. And these delivery tools are then bringing our therapeutic agent towards the target cell. And this is more than just something which is done in the lab. We have approved gene therapies. And it all changed really the way we see this therapy when in 2012, the first market approval in the Western world happened. And this was indeed an in vivo gene therapy for an ultra-rare disease, and we will discuss this later. In the meantime, this is what you see as ex vivo and in vivo market approved drugs and gene therapies, and it's still growing. With this overview, let us now go a little bit more in detail into the different delivery tools and the challenges, as we would like to discuss this today. And we would like, or I would like, to start with an ex vivo approach. And you might remember the word later on of this therapy is Strimvelis, because I really would like to start with adenosine deaminase deficiency as an example of an ex vivo gene therapy approach, and why you will see in a minute. As you might know, adenosine deaminase deficiency is a defect of an enzyme which is causing a severe combined immunodeficiency syndrome. So you are lacking T, B, and NK cells. And this is due to the fact that we generate, due to the lack of the enzyme, a toxic metabolite, and these cells in particular are sensitive to this. What was done, you can do an enzyme replacement therapy, but the more, the therapy which most went forward is a bone marrow transplantation, where you have to find a matched donor and then isolate the hematopoietic stem and progenitor cells, give them to the recipient, and these stem cells will then home to the bone marrow and generate the immune system. However, when you think that you first need to have a donor, and then these cells have to be isolated, giving to the recipient, and they have to match, the more easier idea is, of course, if you can modify the cells of the patients. And here we are right in the fact of the ex vivo gene therapy approaches, where really the idea is to provide functional hematopoietic stem cells, stem and progenitor cells by ex vivo gene therapy. To deliver now the gene, we need tools. And the first tool which was tried in a gene therapy approach were the gamma retroviral vectors. And they sound already like a virus. And indeed, these are viral vectors. And why are we using viruses, which we then engineer to become gene caps or gene taxis? Because viruses have developed very sophisticated and very efficient strategies to deliver genetic material into cells. That is exactly what we require in gene therapy. We need to have our transgene delivered very efficiently into our target cells. And why in this approach gamma retroviral vectors were used? The idea is that these viruses and later on also the vectors which are derived from the viruses can integrate their genetic material into our host genome, meaning that if the cells is dividing, like when you generate a whole immune system from a stem cell, then really the genetic material which you have transferred, your transgene, your therapeutic gene, is also really given to both of the daughter cells. When you have a look what a virus and later on also the viral vector are composed of, it's a protective coat, which is doing the also the communication with the cell, and your genetic payload, so your transgene which you would like to deliver. And just for our first example here, this is a schematic representation of a gamma retroviral vector where you see we call envelope proteins, which do the communication with the cell. And then also the genome in the virus, you have all the information which are virus specific, and the cell later on, the infected cell, will have to generate new viruses. However, if you would like to use viral vectors, then of course we get rid of all the viral open reading frames and put in our transgene expression cassette, so that this is delivered using the same envelope or the same principle as a virus would use to infect the cell. In the first gene therapy, which dates back into 1990 and which was indeed focusing on adenosine deaminase deficiency, this is the reason why I choose this disease to start with our discussion. The gamma retroviral vectors which were there were used in this case to transfer the therapeutic gene into T cells, because at that time, modification of bone marrow cells, or hematopoietic stem and progenitor cells, were very low efficient. The patient in this case was 4 years old, and there was no HLA matched donor available. So when you really look which time we are in, so 1990, you might still know that at that time Margaret Thatcher was Prime Minister of UK, or something else. I just put here some context to show where we are. So that was the time when gene therapy starts. And I found in the internet these pictures. So these are the two girls which were treated with the therapies. And these were the two in 2013. But of course, if you really would like to treat ADH-skip, then you have to go to the stem cells. And that was where a lot of people worked on afterwards. And I just have here one example headed by Alexandra Aiuti and Maria Grazia Roncarolo. In this case, the same vector delivery system was then used to transfer ADA gene into CD34 positive cells. And this is just one picture showing that indeed before the therapy, this was the enzymatic activity. But then after you see 1 year, you really see that now the enzymatic activity is within the patients detectable. In parallel, other therapies were tried, a lot of them also in the area of primary immunodeficiency. And here's another example, because this was indeed the first report of a functional cure in gene therapy. It was also an ex vivo gene therapy. In this case, there was cytokine receptor subunit lacking And also here in this case, the idea was to bring back with the delivery tool, again, a gamma retroviral vector into the hematopoietic stem cells. And this was the first report that it works. It was a study in France. In parallel was a study done in London. And you see really that it is a life-changing event, that patients now having an immune system instead of before really living in a tent. However, and you might remember this, that a few years later after the gene therapy, in some of these patients, leukemia was observed. And this was observed from patients in the Paris study as well as from patients in the London study. And this is explained like the follows. So we need vectors which integrate into the genome if we have cells as a target cells which divide. So therefore integrating vector systems were used. However, the gamma retroviral vectors were not random in how they integrate, because when it is really random, due to the number of genes which we have, the number of genetic information, it's very unlikely that something is disturbed. But if we have certain preferences, as in this case, where we integrate into control region, then of course it could happen then that we activate genes which then in addition with further mutations generate a leukemia. So what is at the moment believed is that we have also the number of vector genomes which were put into the patients, because at that time we thought we have to deliver a lot. The very strong promoter that was used to drive the transgene, then the non-random integration, and then maybe also the disease context really then was resulting in these insertional mutagenesis. So what the gene therapy societies performed then next was that they really optimized the tools that, for example, you changed the control of the transgenes from the very strong viral-specific control elements to internal promoters, and also that you change the tools from something which have certain preferences for control elements for something like the lentiviral vector systems, which are more into the cellular gene regions. And having these new tools in hand, the field moved forward really with the same concept of having an integrating vector, now more safe with the integration profile, because still the hematopoietic stem and progenitor cells are the target, and these, as I told you before, are dividing cells, and we have to integrate our transgene into the host genome. This is a very nice overview which was presented from Alexander Ayuti and his team, looking into 55 trials done or performed between 1995 and 2020. And as you see here in the numbers, they really looked into those studies which were performed, gamma retroviral vectors, next generation gamma retroviral vectors, and lentiviral vectors. And when you have a look at the overall survival, then you really see that these therapies do their job. Indeed, we see a high overall survival and really advantage for the patients. And when you now look at the genotoxic events, then you see that they are caused or have been caused by these first generation gamma retroviral vectors. If we look at the market approval, then with this concept of an ex vivo gene therapy, we have gene therapy on the market for ADA skips. And so you now understand why I really use that example, because it was the first ex vivo gene therapy which got market approval. Others are beta-thal metachromatic leukodystrophy or early cerebral adrenoleukodystrophy. With the same concept of bringing in a gene to repair, functional repair a defect cell, you can also generate living drugs. And this is my second example of ex vivo gene therapy. And this is the example which was becoming famous for this groundbreaking work of Carl June and colleagues, where they isolate T cells, T lymphocytes from the patients, equip these cells with artificial receptor and equipping these cells thereby with the option to identify tumor cells, and then upon recognizing the tumor cells, generate the therapy. We all know this, and I can go over this quite fast. So we have a tumor cell, we have certain changes in the tumor cells which differ, by which the tumor cell differs from a normal cell. And if we are lucky, our T cell can really recognize those and via a kiss of death eliminate the T cells, the tumor cells. However, we all know that the story is not that simple, and that in a lot of cases, these T cells are not recognizing antigens on the surface. So the idea here was now to generate an artificial receptor based on a monoclonal antibody, which really is directing against surface molecules on the tumor cells, and then build in a signal cascade, which then activates upon recognition the T cells. So basically, as you can see here in the scheme, you generate an artificial recognition molecule, which then activates the T cell. And so with what is done is you isolate the T cells from the patients, you do the gene modification, the T cells are then given back to the patients. They are now equipped with a specific recognition receptor, can now identify the tumor cells, and the tumor cells will be lysed. In the meantime, a lot of variants of this basic CAR structure has been done just to make it more efficient, more specific, and so on. But basically, these are the ideas behind those therapies here which have received market approval, and these are all CAR T cells. But they are more than that, really, to fight tumors. You can, instead of using chimeric antigen receptor, you can deliver a whole T cell receptor, really also then addressing intracellular antigens in the tumor cells. Or you can move to other cell types like natural killer cells and also equip those cells with molecules which make them, which empower them as anti-tumor drugs, living drugs. And this is also a very famous application. In this case, it's fibrosis. And this is famous because we move here from an ex vivo approach to an in vivo approach, where LNPs were directed towards CD5 receptors on T cells to deliver in vivo the information of CAR, of generating a CAR in the T cells. And then these T cells, these modified T cells, could fight fibrosis. This brings me to the second arm, and I just check how, it's fine with it, with the time. So I told you we have ex vivo and in vivo gene therapy. And now let us go into in vivo gene therapy for monogenetic diseases. And I introduced to you already the first market approval in Europe for gene therapy, which was the in vivo gene therapy for an ultra-rare disease, which is a lipoprotein lipase deficiency. But you see that it was approved in 2012 and it was withdrawn in 2017, and that is because of that only a single patient was treated after approval. Nevertheless, it was somehow a breakthrough because it really shows that we come from a pure experimental therapy to something which is, in principle available in the clinics. And so I already told you that lipoprotein lipase deficiency is an ultra-rare disease with an incidence of 2 in 100,000 when it comes worldwide. However, we have also some founder effects where we have higher incidence. And basically, the patients suffer from severe hypertriglyceridemia, as you see here. And despite dietary restriction, it's really a very, very severe disease, which is basically without treatment. So the idea here was to use an gain of function variant of the lacking enzyme, the lipoprotein lipase, to inject this into the muscle of the patient via a delivery tool, an adeno-associated viral vector, which we'll look into later on. The second in vivo gene therapy, which got market approval, was a gene therapy for inherited blindness disease. And this disease is caused by a mutation in here, this gene. It results in a loss of the pigment epithelium. And you can imagine if the epithelium is gone, then also you lose just the function of your cones and I'm just missing it. It doesn't matter. So you are really missing the function of your retina and are blind before the age of 30. So here the idea was also to bring the gene back and to bring it back by local delivery. It is a subretinal injection of, again, an adeno-associated virus to bring here back the missing gene. In 2019, this was with a lot of discussions about this therapy, because it is a therapy for patients suffering from spinal muscular atrophy. And these are is the paper which really was making the difference in really showing the results. So again, an AAV9 vector was used. And in this case, this AAV9 vector was bringing in, again, the missing gene, but it was given intravenously into the patients. And the defect really affects motor neurons in the brain. And so this vector is capable of passing the blood-brain barrier. So by an intravenous injection, you are capable of delivering the missing gene into the brain. Further approvals were concerned hemophilia A and B, and you might be familiar with the clotting diseases. Again, it's a single defect, hemophilia A, which is a factor 8 deficiency, hemophilia B, a factor 9 deficiency. And here the target organ is the liver. And again, an AAV vector is used intravenously. It likes to go to the liver, transduces the hepatocyte there in the liver, and provides the missing gene function. And the final gene therapy, which I quickly would like to discuss with you, is again a local administration, which was more recently approved as market approval, where again the missing gene is delivered, in this case, locally into the brain. And all these vectors, and you have heard me already speaking a lot about the AAV vectors, are really delivered, all these in vivo gene therapies, by a single delivery system, the adeno-associated virus vectors, or AAV vectors. And they are based on the adeno-associated virus, which is a dependo-parvovirus. And basically, dependo-parvovirus means that in nature, this tiny little virus here is not capable of generating progeny without the help of other viruses. And basically, it was detected as a virus in adenoviral preparation, so really non-related viruses, as something which was found in these preparations. And later on, it was shown that it is indeed a DNA-containing virus, therefore adenovirus-associated virus, or later shortened to adeno-associated virus. And not only adenovirus can serve as a helper virus helping AAV to generate progeny, but other viruses as well, just those listed here from the herpes virus family. So what is the AAV virus? It's when you look into it, you have a protein capsid, 25 nanometer. You have a single-stranded DNA, which is transported. And basically, this DNA contains a specific structure called inverted terminal repeats, which is specific for the AAV DNA and signals that this DNA and everything which is in between needs to be sorted into this capsid. We have the information of how to replicate the genome and, of course, how to generate the capsid, all encoded in the viral genome, and some helper further proteins. I don't want to go into detail. However, what is most important for you, again, as I showed you in the gamma retroviral setting, you will get rid of all the viral genes and just have the packaging signals here, the ITR left. And instead of having the viral genes, you have your vector genome here, so the information you would like to deliver. And nicely, we have not only one kind of AAV around, but nature provided us with a lot of different AAVs which differ in the receptors they would like to tackle, which means in the kind of cells you can transduce, and you can just pass this genome into the different capsids. Just quickly, frequently you generate these vectors, also the gamma retroviral vectors, by a transfection of cells, where you provide the information of the genome on one plasmid and the information, the viral information, so how to replicate, how to generate the capsid also on another plasmid. And you isolate then from the cells which generate the vectors and purify them by density gradient, affinity chromatography, whatsoever. And then, of course, you have to characterize this preparation. How many particles do I have? How infectious they are, and so forth. So when I tell you with these AAV vectors, we have already 6 market approval drugs, are we done? No, we are not done. And the no is because we use as first generation vectors viruses, or the basis of these first generation vectors are viruses, which are really made by nature and not intended maybe to put genetic information into our target cells. So what we see here is that certain cells are really transduced with low efficiency. These are keratinocytes, important in wound healing approaches. And if you have an AAV vector delivering a GFP into those cells, you don't see any GFP expression. Or if you would like really to transduce a tumor and you use a normal AAV, inject this intravenously, you see everything here lights up with a luciferase gene despite the tumor. And these are things where now the people are working on really to make these vectors more efficient. But to do this, we really have to understand how a virus infects a cell. And let me show this here as an example of an AAV. So if you have a cell on a cover slip and you put an AAV solution on it labeled with a dye, then you can follow how AAV interacts as a cell. And you see that in barely half of the trajectories, you see an interaction with the cell. And then what you see there is somehow a repetitive touching of the vectors to the cell membrane. It's really looking like here the virus is knocking at the door. And in some of these events, you see that it is really causing an entry. In others, it's unsuccessful. And when the 13% of successful entry happens, it's really very fast, so 64 milliseconds. What's happening here is really that the virus, or later on your vector, interacts with receptors on the surface, first with the extracellular matrix, getting a hold to the cell, and then moves forward to secondary receptors. And this interaction with secondary receptors then results, on one hand, in a signaling making the cell permissive for the virus, and on the other hand, also inducing the receptor-mediated endocytosis. AAV then is transported within endosomes along the microtubules into the direction of the nucleus. And you can imagine that when we go from an early to a late endosome and then to a lysosome, when AAV would still be in the endosomal compartment, game would be over. So, but AAV has an enzymatic activity. So when it comes closer to the nucleus and the endosome gets more mature and mature, it puts holes into the endosomal membrane, and it can get out and then deliver its genetic material into the nucleus. And you can imagine with all these different steps, and this is only an example for one of the viral vectors or the viruses, a lot of things can go wrong and or can be inefficient. And here is one example where Mariana wanted to know if we use AAV for the transduction of hepatocytes, and these hepatocytes are from a patient which has been previously infected with hepatitis B virus, is there something going on? And what you can see here nicely, you just have to look at the bars here showing the enzymatic activity of your transgene. If you just have hepatocytes or hepatocytes which have been infected with hepatitis B virus, you see that we have much higher gene expression in those cells which have the hepatitis B virus present. And this is also seen in the mouse model. I'm not going into it, but now why is this the case? Luckily, there are cell lines around which are either hepatoma cells or hepatoma cells with HBV, hepatitis B virus. And so you can step by step understand what is getting better. And what we looked into it here was first, if we infect those two cell lines, do we get the same amount of vectors into the cell? And you see the bars are the same height. Yes, we have the same entry. But then later on in the cell, there is something which is more efficient in those cells which have hepatitis B virus present. And what we figured out was indeed that the intracellular transport was much more pronounced due to the help of one virus protein, the HBX protein from hepatitis B virus. So you see, you could improve the transduction of hepatocytes if you would help the transport from the cell surface to the nucleus. Another example is here. It's a transgenic animal model for hepatocellular carcinoma. And if you inject AAV into these and GFP as a transgene, and first into AAV2, look into the liver, you pretty see that it's not a very efficient transduction. But where the HCC, the carcinoma is, you see a wonderful transduction. And with a lot of experiments ongoing, we decided that here it was not the amount of AAV which could enter into the healthy or the diseased hepatocytes. It was the efficiency by which the DNA could be released from the viral capsid and would be present for serving as template for expression. So you see, there's a lot going on. And the first example here, I would like to show and discuss how we can overcome these barriers. I come back to the keratinocyte as an example of a pre-entry barrier. So we have, you saw this example already. You have primary human keratinocytes. You use an AAV vector delivering GFP, and it is not working. However, if you now modify this capsid in a way that you put onto the surface of the capsid a short peptide which finds a receptor on the surface of the keratinocytes, now the door is open and we get a nice transduction. And it is indeed just a change in the genetic information of how I generate the cap, how the cap looks like. These positions here can nicely fit to the receptor on the surface, and if you have the right peptide there and you know the receptor on the surface to which it can bind, it's a wonderful option to improve the efficacy and overcome entry barriers. And since you are changing now the capsid, you might even generate variants which, although the patient has pre-existing antibodies against this vector system, as you see here, when we have AAV2, we transduce the cell, all the cells are green. However, if you have serum of a patient which has neutralizing antibodies, we don't get a transduction. However, if we now modify the capsid, it can enter because the antibodies are not recognizing this region anymore. But can we also address the issue of being really precise that we can really off-target redeliver something to our target cells. And don't be afraid of this example here. This is a wonderful collaboration done with Christian Buchholz at Paul-Ehrlich-Institut in Langen. This is just a scheme to show how the modification was done. So what here was done was we used a design anchoring protein, which is somehow you can compare it with an antibody for providing specificity of your target molecule. And these DARPins can be generated artificially so that you can really, engineer these molecules that they find your target molecule on the surface. Indeed, it was fused to one of the capsid proteins so that it is now on the capsid surface. So you might remember this picture. You use AV serotype 2, inject this into a mouse having a tumor, which on the tumor is a molecule called HER2/neu on the surface. And if you look where the cells are in the body of the mouse that were transduced, you see nicely here images, but no transduction of the tumor. However, if you now have engineered this particle, this AV particle with a DARPin recognizing specifically human HER2/neu and sends this into the same way, you see that only the tumor lights up, because this specific DARPin recognizing a specific molecule only present on our tumor cells. And this works also in the disseminated tumor. This is, of course, an artificial system, but it shows you if you have the right combination of a receptor and a targeting molecule, what you clearly can achieve. However, if we don't know the receptor, if we don't know the barrier, what can we do? In this case, we use what nature is doing. We use a library approach, evolution. So you recognize already this AV particle, and you know from the keratinocyte work that you can put here ligands which find a specific receptor on the surface. However, if I don't know the receptor to target, I can generate a library of AV particles which differ. Here, this one differs from this one in the peptide that is seen there. And with this library, you now can do selections in vivo in your transgenic mouse model, for example, or non-human primates, and identify from this library those particles which indeed can, for example, via your intravenous route, reach, for example, your muscle cells. And you collect those virions, generate a new library, and do another round of selection. And this is one example that it works quite nicely in collaboration with Stelios Michakoulis from the LMU Munich, where selections were done in the mouse, where the library was injected into the tail vein of a mouse, and the virions were collected from the retina. And we were ending up with virions which really accumulated there, and they were then further investigated as vectors in vivo and compared to where we started off. So with the purpose to generate vectors which are more efficient in vivo transduction of the retina. These vectors, this is where we started off, AAV2 expressing GFP after intravitreous injection. This is what you get after 1 week. However, these variants which were coming out of the selection and which really differed only by these small insertions in these green dots, which I showed you, you see that they're much more efficient and brightly spread into the retina. And it's not just we selected them in the mouse and then, okay, what is going on then in other species, but it's also working in the dog, in non-human primates, or here Stelios also tested them in explants. I cannot show you the second example. This concerns the liver, but in principle, also here you see that you really end up with important new vectors which transduce the hepatocytes of the liver much more efficient than where you start off. And these really transduce murine and human hepatocytes alike. So you can really do by these capsid engineering overcome these problems, these challenges of target transduction, low transduction efficacy, pre-existing immunity, really, as I nicely showed you, hopefully nicely showed you with the examples we discussed. So coming back to 1990, so this was the first human gene therapy done as an ex vivo gene therapy using gamma retroviral vectors and T cells. We are ending up at the moment here with gene therapies which are on the market and which is really some that you see that a lot of is ongoing is this expectation here that FDA expects from 2025 onwards 20 new market approvals per year. So you can imagine what is on the market. However, I think it's not only that we have to bring it to the market, develop these therapies to our patients in need, but there's a lot which is ongoing afterwards. And I will just briefly quick through it, and we can go later back when we discuss, I have the Q&A, because really we have to be fast. Time is everything for the patients. We have really to ensure that the therapy after being market approved remains accessible. We have to work on reducing, of course, the cost for treatment. We have to really come up with good solution for the vector production, but also to move on and improve current therapies, availability of real-world data are there. We have to go back and improve what has been done. And there's a lot which we have also to do, training our next generation scientists, physicians. We have to speak with lay public stakeholders and so on, and of course engage quite early with regulatory bodies. By this, and before starting the Q&A, I really would like to acknowledge a wonderful team of people. Here you see the team of the experimental hematology, so the whole institute. And I really would like to in particular thank my team listed here, as we are part of this wonderful Institute for Experimental Hematology. And this work can only be done with a lot of collaboration partners. It's a team effort, and I'm really grateful to each of these collaborations and that I was allowed to really show the results of these wonderful collaborations. Thank you very much.
Thank you very much, Hildegard. That was a very impressive and comprehensive explanation of a team that is complicated for me, and I know that there are in the audience people who totally understand you. So I'm in a bit of a trouble because there are no questions yet in the Q&A, and I need to pretend to know something about this and put you some questions. So, but that's what usually happens in these webinars. So you mentioned many of the troubles that we face still today with these kind of therapies. And one of them is the limited space for carrying the information. And I know that in the past that was a limiting factor for certain treatments, because you would need more space to transfect everything that you needed in a certain problem or in a certain genetic defect. So how is that evolving, and what are your perspectives in terms of optimizing that capacity of carrying material?
Yeah, so this is a very, very important point. So of course, when you have your delivery tool, you really are bound how much genetic information your delivery tool can provide and deliver, of course. And first of all, and therefore I also included as the gamma retroviral vectors, the lentiviral vectors. I mentioned briefly the alpha, I mentioned also the LNPs and so on. I think the most important thing is really that we have to be aware that we have different delivery tools around. And that there is a lot experience now with the first generation. And now we start to engineer and really improve what we have from the first generation. And with regard to the size, on one hand, of course, you can try to shrinken your transgene to a format that fits, where you really engineer your therapeutic agent in the way that you only have those functions left which are required for functional cure. Or what you also can do is that you come up with strategies to increase the coding capacity, let's say. And there are really, for AAV, for example, there are approaches around called dual AAV approaches, where you bring in different parts of the genes via two different AAVs into the same cell and make sure to a lot of tricks that these two parts of the genes are then recombined and either on a DNA, mRNA, or protein level to really provide them the function. Or as I said, you use another delivery tool which has more capacity.
Excellent. And what about the immunogenicity issues that are sometimes mentioned regarding these therapies? Because in some cases, you're not looking at a once-in-a-lifetime administration, isn't it?
So these therapies are intended to be once in a life, because the issue is indeed that you generate with viral vectors, an immune response. And due to the presence then of neutralizing antibodies, you indeed cannot go back with the same kind of delivery tool. And as you might know, when we have a market-approved drug, we cannot just change the delivery tool. So we are bound to exactly the same delivery tool. So therefore, with regards to immune response, first of all, of course, the patients have to be monitored for the presence of neutralizing antibodies against your delivery tool. And if they do have antibody titers which are above the respective thresholds which has been set, then they will not be eligible to receive the tools just because, or this therapy, just because it would on one hand not be effective due to the neutralizing antibodies, and maybe you even put the patients into risk because the immune system is maybe already alarmed. On the other hand, of course, we also have to think in gene therapy that also the transgene could generate an immune response because, of course, if this transgene as a protein has never been seen by the immune system before, then it would be something which is foreign, and also there our immune system could react. So the immune system is really something which is an important issue to consider on one hand, because it maybe interferes with your delivery tool or generates an immune response because your immune system recognizes your delivery tool. And on the other hand, also concerning the transgene.
Thank you. I was hoping that by now some geneticists and experts were popping questions on the chat, but I need to continue. So I'm sorry for probably delivering you some silly questions. So the next one has to do with the bottleneck of production of these vectors that you mentioned in your talk. And I would add that they are very complex technologically, and I imagine that it may take many months, or I don't know what is the time frame to produce those vectors. And the other thing is that it implies a lot of costs related to environmental protection, and it has also an impact of the final cost of the treatment. And what do you think is going to happen in the future? Is there any rationalization and optimization pathway that we can follow to improve, on one hand, the production of the vectors, and on the other hand, keep them safe and not too expensive?
Yeah, I think there's a lot ongoing. As you said, generating these tools independent which kind of delivery tool you have to focus on. It's a very complex system. On one hand, because you of course have to ensure that only your viral vectors are generated, so that you, for example, in the virus field or viral vector field, are not generating a virus. Of course, you will only generate your vectors. You have to purify them. The incoming material is very expensive. The production per se is very expensive because, of course, it has to be ultra pure in the process. So you are because it will be later given to a patient. Yeah. So the production is really a science for itself. And then, of course, and this is what I wanted to discuss with my second part, with regard the efficiency and the specificity. So on one hand, we have this complicated production system where a lot is ongoing to simplify this. But on the other hand, also the amount of vectors which we have to provide. So if we now increase the efficacy of the vector, being that that you just need less particles for per cell who really give your transgene to the cell, or when you think about in vivo, that you are indeed specific and that you are not losing your vectors and off target, you can also reduce the dose, which on one hand will reduce the immune response, which you might elicit. And on the other hand, of course, the amount of vector you have to give. So on one hand, it's really to simplify and automatize the production process per se, make it more efficient and more easy. And on the other hand, of course, to reduce the dose which you have to give to the patient by making the delivery tools more efficient.
Excellent. And so in terms of the safety regarding cancer growth and incidence of neoplasia in treated patients, you've shown that for most of the patients that are well chosen for the treatments, you have, on the contrary, a very good reduction of mortality, etc. But on the long term, what do you think real world data is bound to show? Do you have any idea about what are the possible consequences in 20 or 30 years from the beginning of the first treatments?
I think, I mean, the number of patients who have been treated so far is still comparable low, right? And the patients are monitored. So to predict something or assume something, it cannot be done, of course. But what I wanted to show you with this, also with this presentation, is that safety is an absolute first, how is it called, first choice, yeah, for us. So which means that really the vectors are improved constantly with regard to safety. So that you, if it is not required, you are going for a non-integrating system instead of an integrating system. Yeah, just because you don't want to really have the danger of manipulating your host genome. On the other hand, also, if you think now about gene editing, where you now have not to rely on a random integration, but could make a precise cut and put something into a safe harbor, this is also something which is a plus with regard to safety. Yeah. Yeah.
So with these hopeful thoughts and great and shared wish to help our patients and develop more efficient and more and therapies for more diseases, we are going to close. I'm sorry that I was the only one putting you questions, and I want to thank you very much for your very nice and very clear presentation, and I'm certain that we'll have an opportunity down the road to talk again and to meet again. So thank you very much, Professor Büning, and thank you to all that have attended. Please give us feedback after you disconnect from the webinar, if you don't mind, and see you next week. Bye now.
Thank you very much.